A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speakers, Dr. Van Denham and Dr. Fanchi, will discuss their experience with setting up adaptive immune repertoire sequencing for antibodies. Please note, this webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I now turn the show over to our first speaker.
All right, Jan, thanks for the introduction. Today I'd like to tell you something about the work we've done to set up a repertoire sequencing assay, specifically for antibodies. The assay is applicable more broadly than just antibodies, but that will be the focus for today. In the first part, I'll be talking a little bit about some of the lab and the simulation approaches that we've used. And in the second part, my colleague Lorenzo will take you through a sneak preview of our new processing software. All right, so why are we actually looking at repertoire sequencing to investigate immune repertoires? Well, it's actually a very powerful tool. Essentially, it's the quantification of the T and B cell receptors that can be used for all sorts of purposes. Common applications include immunotherapy monitoring or treatment monitoring, tracking vaccine responses, MRD, but also drug discovery. And specifically for antibody candidate selection, it's very useful. In earlier days, people would frequently use Sanger sequencing. It's still used, but more and more there's a switch towards high throughput sequencing, high throughput repertoire sequencing, which allows you to quantify far more candidates and do a far more in-depth screening of your antibody candidate pool. So that's why we also decided to set up repertoire sequencing. Now, originally, ENPICOM is actually well, of course, it's good to mention that this trend is set to continue, and indeed, there's going to be more applications of repertoire sequencing in general in the future. The origins of ENPICOM is actually that it's a software company, so we focus on the processing and analysis of software for repertoire sequencing. So we have a cloud-based platform called the IGX Platform that does analysis and is specifically tailored towards finding and helping selecting the right antibodies. You can, of course, add and remove data from this platform and interact with several databases or add extra information. And later on, Lorenzo will specifically be saying something about IGX Profile, our processing app. But for now, I'd like to take you through some of the work we've done to set up a full service offering to include both the wet and the dry lab. So setting up Rep-Seq involves going all the way from sample all the way to discovery. So there's some sample prep, there's specific sequencing protocols, there's specific processing protocols. And in the end, you want to go all the way from sample to discovery. But since we are a software company, we teamed up with Viroclinics DDL, which is a part of Cerba these days. And together, we decided to set up a full service repertoire sequencing offering that would, do all the things to go from sample all the way to discovery. In order to set this up, one of the first things we did, of course, is to look at, well, what specific components do we need? And in the end, we settled on an amplification kit by Takara. It's a 5' raise kit. So over here, you can see some of the different approaches that can be used for repertoire amplification. There's both RNA and DNA protocols available, but the one we've selected is a 5' raise protocol, which is shown on the left. And this protocol has the advantage that it does relatively unbiased sampling of the or amplification of the repertoire, and can also be used in Illumina sequencers. At the bottom, you see the construct or the structure of the read that we get out. Well, we try to apply this, and our first results looks quite good. And obtaining bands of the correct size, things like that. The results all in all look very promising. So one specific feature that is part of the Takara kit is the unique molecular identifier. For those of you who are not familiar with this concept, a unique molecular identifier is a small barcode, which is, in the case of Takara, 12 nucleotides long, and it is used to tag all the separate RNA templates, and thereby you're able to correct for amplification biases that occur during the amplification and the sequencing. So by adding this specific barcode, what can be done during the processing stage, which you can see on the right, is that you use all the different replicates that have been generated during the amplification sequencing phases, and then fuse them back together to try and reconstruct the original number of templates and also the original sequence. So the little crosses represent sequencing errors, and these can also be corrected because you can do basically a consensus assembly, and this then allows you to get back the sequence that was the sequence of the original template. So in order to check whether the error correction went correctly, one of the things we can do is to look at the N bases. Of course, repertoires are highly diverse, and they can contain somatic hypermutations and all sorts of other features. So typically we don't know the original sequence of say a generic repertoire, but by looking at the N bases, we can actually see if the error correction works. And that's what you see in this case. So the bottom row, you can see that the two left panels have higher levels of errors or of N's than the two right. And this is the effect of error correction using UMIs. So you can show here that, that this is actually effective for this particular assay. So how many of these UMI would you like? This is, of course, always a sort of a distribution, and you can see the distribution in the slide here. You try and aim for a good number of reads per UMI group, and we typically like to use about 5, or we aim for 5. And the reason for that is that 5 allows you to create a good consensus sequence based on your UMI groups. If you have very few, say 1 or 2, it's very difficult to create a consensus sequence because you have very little information. And on the other hand, if you have too many, it's sort of a waste of sequencing space. So that's why we try to aim for what we call the sweet spot of about 5 reads per UMI. In this particular plot, it's also, it runs from 2 to 25 plus. And what we see often, and this is also a feature of repertoires, is that the singletons, so single templates with a single UMI barcode, actually make up the majority of the data. And this is something we see frequently and something we try to reduce, but this is actually something that, that is quite well known in repertoires. In fact, when we try to optimize our assay a little, we noted that by using different amounts of starting material, as you can see here in the slide, the ratio between singletons, also having a very high number of say 25 plus category UMIs, that shifted quite a bit. So you can see that these 5 panels represent 100, 200, 400, and 800 nanograms of starting material. And in this case, you can see that low amounts don't really work well. And the best one is the one I showed in the previous slide, which is the bottom left one. And there you see the number of singletons is relatively low. We're relatively close to the 5 sweet spot. All right, another value we look at, another metric we look at is something we call receptor recovery or receptor recovery rate. This is the number of reads from the Illumina sequencer that are used to construct your repertoire. So every read is an amplification of a receptor, but of course there can be misreads, there can be junk in your sample, something can go wrong with the amplification. There's all sorts of things that can lead to a read that does not contain a receptor that can be identified. In this particular graph, it's not the data that we generated in this particular project. It's an example data set that has quite a lot of junk in it, just for illustration purposes. And in this particular data set, we had to perform trimming, we had to perform UMI processing, and after that, the receptor extraction. The receptor trimming was needed because there was quite a number of adapters present, and UMI processing was there. Of course, there was UMIs there, so that was good to try and do the error correction. But what you can see here is that a large number of reads had to be removed because of the cleaning up process. And if you just use all the cleaning up steps as you would normally, you see that you have less than 10% of your data left. That's the far right. You see that the percentage of reads that's still present in your final repertoire is all below, generally below 10%. So this is not a very a good data set, one could say. With the approach that we've set up with DDL, we typically get receptor recovery rates of about 85%. So 85% of the reads contribute to the construction of a clonotype. All right, so what about Rep-Seq replicability or reproducibility? So before we start looking at that, I think it's good to remember what we are exactly measuring. So we know that the number of potential immune receptors is about order of magnitude 10 to the 15 or larger. The number of T and B cells in the human body is about 10 to the 12. It means that if all things were equal, so all clonotypes are equal, every B and every T cell would have a unique receptor, or could potentially have a unique receptor. The number of lymphocytes in a normal blood sample is about 10 to the 7. So that's, of course, a very small part of this whole big pool that we have in the body. And this essentially leads to the expectation that the number of cells per clonotype in your sample is 1, or order of magnitude 1. So this means that if you want to look for overlap, you're actually looking for very small clonotypes that, unless they've been expanded, clonotypes are expected to have, say, a single cell in a single sample. And this can vary from sample to sample, which means that it's quite hard to find the same samples or the same clones in different samples. And that's what we've done here. We've taken a look at the biological, two biological replicates. So it's two separate, it's a single blood draw which has been split and then processed separately. So the cells in the sample are different. And there you see indeed that although there is some concurrence that you can see around the diagonal, you see there's lots of points to the sides of the plot. And that means that these clonotypes are found in only one and not in the other. If we look at technical replications, so these are two samples sequenced, but they are from the same RNA source, so from the same set of cells whereby the RNA isolation has been done in one go, you see that the concurrence is actually quite good. And you see that the majority of the points really fall on the diagonal. But this illustrates the point that there's quite a bit of difference between biological and technical replication here. And that is due to the large number of receptors that we have and the large variability of receptors that T and B cells carry. So notwithstanding the difficulty of quantifying overlap, what we observed is also here that if you have different amounts of starting material, that this influences the overlap that you find. So if you start off with say 50 nanograms of starting material, you see that the overlap is quite small. And for quantifying the overlap here, we use the Ruzicka index, which is a sort of a weighted Jacquard index, and essentially tries to see on a read level, do I see the same receptor in the two different samples. So it weighs in the size of the clonotype that you identify. So large clonotypes have a larger weight than singletons. And what you can see from this plot is that the more you progress to the upper right corner, you see that this is the overlap between samples that were generated from more starting material. you see that the overlap between these samples slowly increases from about 0.2 in the bottom left to about 0.6 or 0.7 in the top right corner. And this means that the amount of starting material will definitely have a big impact on the overlap you find between your replicates. So this is good to remember. And we thought that for the best reproducibility in our hands, with our assay, about 400 nanograms would be about appropriate. This is also about the same amount that gives a good UMI to read ratio. All right, so in order to validate our setup a little bit more, we decided to use some synthetic clones to really track what's going on in the amplification and in the sequencing. So in the past, people have used these sort of benchmarks and have used cell lines to do this. There are several publications out about this. But we opted for using synthetic RNA, and this would basically generate synthetic clones. So we designed very specific sequences with existing V genes, with artificial CD3s. And the advantage of this, of course, is that we know exactly what the input is. There is no margin for error there. And the also big advantage is, and rather than using simulated data, which I will talk about later, this is actually real sequencing. So this is actually real data. So you can actually look at the real, the real differences and the real effects on that your assay has on synthetic constructs. So in order to do this, we designed several spike-ins, and we designed them such that we have a different ratio, so twofold changes over 5 spike-ins. And also we used a few spike-ins that differed in length. The reason for doing this is that this is a 5' race protocol, and it's thought that 5' race protocols can have a length bias. And since we're already pushing the limits of the MiSeq sequencer, so the 600 base pair length we do really need, we decided it would be good to also test that. So for the different concentrations of spike-ins first, the twofold changes. So we spike these in a few different fractions, so different amount of nanogram as compared to the original sample. So this is done in a PBMC background. Spike-ins were then processed, isolated based on their sequence, and from there we can quantify them. So the lowest amount of spike-in the end was about 100 templates at the bottom left corner. And there was, of course, the higher numbers were really quite high. We also included 1 or 2 samples of spike-in only, just to see how that would do. What you see here is that all of the spike-ins could be very well quantified. They're quite reproducible. They're more or less all well in the diagonal, though there's a bit of a peak in the green line, of course. If we look at the regression slope, it's about 0.96. So we also see that the quantification is very close to what the actual amount is if we look at the number of copies that we have available in the sample. So this works quite well. If we now look at the length spike-ins, we see that indeed we see a length bias, and we have quite a large range of this, and it's a little bit more variable than the amounts spike-ins that I showed. But you very clearly see that if you look at the clone counts for the larger CDR3 lengths are quite a bit lower than those for the shorter CDR3 lengths. Of course, the amounts have all been normalized, so all the spike-ins are included or are spiked in an equal number of copies. So in principle, we should see a straight line or dots that align horizontally, but we clearly see a downward trend. Also the difference between, say, an 8 length CDR3 length and the 32 is actually quite large. It can be 2 or 3-fold even. So this is definitely something we're looking further into to see whether we can we should take this into account. If you want to compare clonotypes say, over time or something like that. This length bias doesn't really need to be a problem, because if you want to find the same clonotype over again, of course, the length is the same. So there's no bias in comparing clonotypes of the same length. It's just that if you assume they're all expressed equally, then you should be aware that the long CD03 lengths will be harder to detect than short CD03 lengths. All right, so this is for the first part. I've told you a little bit about the wet lab stuff, the full service thing that we've been developing. So in summary, it's a high quality Rep-Seq setup. We see, of course, the sampling effects, but the quantification is accurate. We do see a length bias, and overall we get a very high receptor recovery rate. So a large part of the data contributes to the repertoire. So that was part of the journey that we had for developing the wet lab stuff. Of course, being a software company, being bioinformaticians, we also very much like to do simulations to be in full control of everything that happens. And for that, we also have been developing a realistic simulated data framework. The idea here is to have a fixed input so that we have a very good way of benchmarking our new tools and other tools, so that we can be certain that what we process is doing it exactly right. For the simulation framework, of course, we use the insights we obtained in the wet lab for setting up the wet lab procedures. But we also made use of some existing tools, notably ImmuneSim. So ImmuneSim is an immune simulator. It has been built and published by the Greif Lab some time ago. And it simulates artificial receptors, essentially by reassorting existing receptors and existing annotated receptors, as you can see in the picture on the right. It can also simulate somatic hypermutations anywhere in the receptor, and has some advanced features that we don't need for this particular problem. One of the enhancements that we added ourselves is that we are now able to use any pre-formatted germline reference. So we can replace the reference that ImmuneSim uses by default by one, for instance, from another species, or something that is very specific to a, say, antibody screening construct, something like that. This can now be done. And this we use as the basis, or as the raw substrate for simulating. So the next step is to simulate the amplification part. And for this, we built a small application ourselves, which we called AmpliconsSim. It converts receptor sequences to amplicons, and it can, for instance, simulate barcode inclusion, scaffolding, but also UMI to try and simulate this part as well. We can simulate different constructs such as heavy and light chain constructs that you would find in SCFE data, which is also antibody specific. And sometimes what happens is that this amplification includes a certain bias because certain receptors are missed, or there's no capacity to do this. So if there can also be a sampling effect there if that is required. So this amplicon simulated data is then turned into actual FastQ files by feeding it into a tool called InSilicoSec. This is a well-known simulator for instance, HiSeq, MiSeq, and also NovaSeq data. So essentially, you put in a sequence, it simulates the type of sequencing errors that one might expect for these machines. It generates the threat scores associated with those. And it outputs them as FastQ files. So we were working with the original author of this project to extend it a little bit, because originally it was built for metagenomics data, and we've now also made it such that it does amplicon data, and we're working on a few other enhancements together with the original author to try and make it more useful for repertoire sequencing. So, using all these three things together, we now have a simulation framework that really allows us to go from a fixed input all the way to comparing it to the output, which means we can see exactly and validate exactly all the processing steps that are required and make sure that they're on par and that we don't have any oversights or any artifacts that we cannot address. All right, so this is the second summary before I hand over to Lorenzo. So what I just showed you is, in addition to the full service setup, we have a simulation setup which aims to produce realistic data. It includes repertoire generation, amplification, and regeneration. The tools that we use are partially published, and the aim is, and I think we've achieved that, to go from a controlled input to a controlled output that we use for testing. With this, I'd like to hand over to Lorenzo.
All right, thanks a lot, Henk-Jan. Yeah, thanks for explaining all the details that go into setting up an accurate and most of all reliable repertoire sequencing service. Of course, as we already mentioned, next to the sequencing itself, the processing of the raw sequencing data is of paramount importance to a proper downstream analysis. And this is actually where the IGX platform picks up. So as you can see in this overview, basically everything up to the raw sequencing data coming off of the sequencer is what Henk-Jan just explained in his presentation. And the IGX platform picks up right after that. So the IGX platform can process repertoire sequencing data. It can integrate that sequencing data with any additional metadata that you might have generated in the lab. And finally, it allows, through various applications that we developed, to select better antibody candidates. So today, I want to give you a sneak preview or a sneak peek at the next generation of our clone profiling and annotation application, which is called IGX Profile. IGX Profile is part of the data processing apps. So on the left side of this figure, the data processing apps of the IGX platform, and it works by ingesting raw sequence data, such as FASTA data, AB1 data, or FASTQs coming off of any Illumina machine, and delivering fully annotated clones that are stored in the IGX platform. So basically in the data handling part of the platform. And subsequently, you can analyze these clones using any of our downstream applications. You see a handful of them on the right. And these applications are specifically tailored for the various antibody discovery workflows, such as, for example, hybridoma technologies or display technology approaches. Now, for IGX Profile, our goal was to create an application that can basically be used by any researcher. You don't need any knowledge of how to code or any of these things. No matter the complexity of your amplicon structure or the sequencing technology that you want to use. And some of the key features that we developed for IGX Profile are the use of templates. So to make sure that you can spend your time on actually analyzing your data rather than setting up various configurations using command line interfaces or non-maintained academic softwares. We provide fully customizable templates with which you can easily build any amplicon structure that you might have. So we have, to achieve this, built comprehensive support for various UMIs, also for barcodes that are often used to pair chains. So for example, in 10x based technologies, but also at the same time for linkers. So you might, for example, use SCFV constructs that link a heavy and a light chain in the same amplicon. And these things are basically a breeze to set up in IGX Profile. And while we know that people still, of course, use single-chain sequencing, many people are now opting these days to go for technologies that actually capture chain-pairing information as well. So we've made sure to build in support for pair-chain sequencing methods, such as various plate-based methods, but also droplet-based technologies, such as, for example, the 10x Genomics and Berkeley Light platforms. To ensure, of course, that your data is of the highest quality, we've also built a proprietary algorithm for UMI error correction and eventually consensus generation. And on top of that, we also provide extensive control over filtering of your outputs. So here you can think of things like filtering out non-productive rearrangements, setting thresholds based on read counts, or, for example, UMIs, a number of reads that back your UMIs. And this goes all the way down to granular control over ensuring, for example, that your single-cell data adheres to a strict definition of a cell. So we can, for example, make sure that every clone that we output contains at least a heavy and an associated light chain. Of course, as with all apps on the IGX platform, we've paid particular attention to providing an intuitive user interface and an intuitive experience, while at the same time also allowing for a high throughput configuration. So we can easily set up analyses of hundreds of input files at once. And finally, of course, we've also made sure that we can support the latest generation of sequencing systems such as Illumina's NovaSeq. Now, these are some of the key features that we developed for IGX Profile, and I would love to show you actually a sneak peek at the UI for Profile. So once you start IGX Profile, you land on this overview page where you can see two main buttons. The one on the left is used to start your analysis. You can start your analysis after you've set up two different kinds of profiles. So we have, two different kinds of templates. We have sequence templates and we have quality control templates. So if we were to go into the template configuration page, this is where we're at now. So you can see that on the top of the mockup we see two tabs: the sequence tab and the quality control tab. So you can see the sequence templates that you've built or the QC templates respectively. You can create and store as many different templates as you might like. You can see here that we've generated templates for 10x based amplicons, an SCFV that contains a heavy, a linker, and a light, or a plate-based template. What you can also appreciate is that for each template, we give you a visual preview. So you can see that many of these templates are pair-chained, as indicated by the double arrows on the left and the right of the amplicon. You can, of course, rename. You can give each template a name that you might want, and we'll also provide the ability to share templates with other users within your organization. So you can either keep your templates private, or you can have them used by others in your organization. And yeah, with that, I would actually like to show you what our template or sequence template builder looks like. So if we were to open the 10x protocol, we actually now head into the sequence template builder. If you focus your attention on the top, you can see our control bar. So in the control bar, you can set, for example, the endness of your sequence, of your sequence system. So in this particular case, we are configuring a paired end construct. And if you focus your attention on the center part, you can see all the different elements that that you can use to compose your amplicon structure. So the way that this would work is you can actually click and drag any of these elements, such as, for example, an IG block, a TR block for antibody or TCR receptor data, respectively. You can see a barcode block, a UMI block, and linker blocks. And you can click and drag any of these blocks onto the canvas that you can see in the lower half of the screenshot, to compose any amplicon structure that you might want. If you click on any of these blocks, you actually open the configuration pane, and here you can set a detailed configuration for any of these blocks. Now, as I mentioned, we don't just have sequence templates that define your amplicon structure, but we also have quality control templates. So here you can see an example of a QC template where you can see that we have various options that you can set, like requiring productive rearrangement, various different read count thresholds, base quality thresholds, or even the ability to discard sequences that contain specific motifs, such as, for example, liabilities that you might not want within your sequence. And similar to the sequence templates, also for QC templates, you can make as many different ones as you might want. You could have a template that defines a very strict level of quality, where, for example, you would want the full VDJ sequence to be productive. You want to have at least, I don't know, 5 copies or 5 observations of your sequence, or you can configure a very lenient quality control template. So once you've set up both a sequence template and a QC template, you can go into the analysis configuration part of the application, and that's what you're seeing right here. So in this particular example, you can see a card in the center of your view, and this card actually configures the analysis for 2 input files. You can see them on the right hand or the right half of this card. So this is a 10x output. And basically how this works is you configure the analysis through all of these controls on the right. So you can see that there are buttons to assign a sequence template to these inputs, the orientation, which of course, if you're using an Illumina machine, requires you to specify the forward and the reverse reads. There's also options for chain pairing, which of course for Illumina, which of course for a 10x based setup is conferred through the sequence level barcode. But you can also use other ways in which to define chain pairing. And if we go further down, you can see that we can configure the quality control template, of course, the organism with which you want to annotate your sequences. And there will be various options for picking different references. And after you've performed all this configuration, you see the result of your configuration on the right. So in this particular example, we've configured a 10x sequence template that will yield an output collection of clones that contain both a heavy and a light sequence that are grouped together by the barcode that is encoded in the read. So with that, I hope I could give you a nice impression or a first impression of our next generation clone annotation app called IGX Profile. IGX Profile is coming very soon, in about a month from now, and it will support all the different features that I've highlighted previously, such as a wide range of paired chain sequencing methods. Of course, through the sequence templates, we support any amplicon structure that you might have developed in the lab. Extensive QC options, a flexible and intuitive user interface, as I've hopefully managed to convince you of. We'll have proprietary algorithms for UMI error correction and consensus generation. And of course, all of this scales to support the latest Illumina or other technologies. And with that said, I would like to thank you for your attention, and yeah, we're happy to take any questions. which I believe will happen through the Q&A pane, if I'm not mistaken.
Here we go. I'm back. We do have a few questions. Let's start with the first one, which came in relatively early. The question is two parts. First, when you use UMIs, do you lose depth sequencing? Followed by the question, how do you deal with the frequencies of clonal groups?
Right. To answer the first question, do you lose sequencing depth? That depends on how you mix. So it depends on the ratio of UMI with respect to reads. So if you decide to put in little input material, you'll have lots of reads in every UMI group, and you will effectively be losing some depth. But that's the price you pay for the added accuracy that you get for having more than one copy, so that you can reconstruct your template. There is always the option of not analyzing your sample with use of UMI. So you can simply ignore it. You can analyze your data as though there's no UMI there. So that is always an option. But that is sort of the balance that you're trying to strike there. And then the second part of the question, I believe, was how do you deal with the frequency of clonal groups? I'm not quite sure what is meant by that, to be honest. So maybe a person can clarify and we can move to the next question.
Yes, that was going to be my suggestion. Next question. How do you achieve 600 base pair reads on the MiSeq?
Yes, so the 600 base pairs is actually 2 times 300. So it's paired end sequencing, and each read is 300 long. So in total is 600. And that is there are special kits for that on the MiSeq. So this is it's not used in too many other applications, but it is a standard option for the MiSeq. So that's you can just order it.
And the next question is your pipeline adapted or transferable to livestock species?
Yes. In principle, the setup and also the IGX platform is species agnostic. There is one thing to mind, or actually two things. First of all, if the species in question makes use of gene conversion, there's a few species that do. So they sort of interweave their genes, for instance, make use of 2 different feed genes at the same time by changing them halfway through. That is difficult to analyze currently. And the second is that there is also the need for a good reference. So if there is a reference, so germline reference, feed gene references, for instance, and J gene references, if those are available for your livestock species, then yes, then it's certainly possible.
Thanks for that response. The next question is, how much will IGX profile cost to use?
Yeah, I can take this one. So maybe just a little information before that. So Profile is part of the IGX platform. This is a cloud-based software as a service platform that for which there's a licensing fee, or a licensing fee for which there is a fee. Yeah, for any specific sales-related inquiries, I would be happy to put you in touch with one of our sales executives. Maybe we can. I don't know if there's an opportunity to connect outside of this part, or outside of the webinar. But otherwise, feel free to head to www.empicom.com, and it will also be easy to get in touch with one of our sales reps.
Are there any further questions from the audience? Opportunity is going, almost gone. One more last second. Okay, I think we have gotten all of our questions. So In concluding this webinar, I'd like to thank our speakers for providing insights into setting up adaptive immune receptor sequencing for antibodies. And I thank you very much for joining the webinar today. An on-demand version will be made available within a day. I'll send a link to it via email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again and have a great rest of your day.